All right, sorry those who are just watching on YouTube. I'm going to go back really quick. First slide, we talked about oxaloacetate. Key takeaway here is that we're going to be talking about making glucose, and we're going to focus on pyruvate to glucose, but all intermediates have to go to oxaloacetate. All right. So the creation of glucose uses a lot of the same enzymes as glycolysis does, except for 3 enzymes that we have talked about before: hexokinase, phosphofructokinase, and pyruvate kinase. These 3 enzymes have a very large delta G, which makes them irreversible. Therefore, if we want to go and make glucose, we need different enzymes. So instead of hexokinase, we use glucose 6-phosphatase to remove our phosphate. Remember, phosphatase a lot of the times means to remove a phosphate. Instead of phosphofructokinase, we have fructose bisphosphatase. Again, since we're going reverse, we are removing our phosphate. So that's why the bisphosphatase comes in. And then at the very start of our pathway, we have 2 enzymes. We have pyruvate carboxylase to make oxaloacetate, then PEPCK, or PEP carboxykinase, to make PEP. Otherwise, it is the exact same pathway. So just 3 different steps here. So let's start from the bottom, starting from pyruvate, working our way up. So we need to convert pyruvate to PEP via 2 steps. We need energy going into this. So the arrow down here is cutting it off, but you have ATP going in to our cycle with carbon dioxide as well. And we get our carbon dioxide by our acidic form of carbon dioxide here. In biochemistry 1, we talked about this molecule. It's your blood buffer. We can convert that to carbon dioxide, no problem. So what you do is that you make oxaloacetate, and the only difference between oxaloacetate and pyruvate is carbon dioxide. So that's kind of thing easy to remember there, right? Pyruvate, oxaloacetate, different by 1 CO2. So our first step is to add this CO2 back to make oxaloacetate. That costs ATP because we're making a carbon-carbon bond. Then we have to spend more energy, and this time in the form of a GTP. And what PEP carboxykinase does, remember, kinase, a kinase adds a phosphate. So PEP carboxykinase adds a phosphate from GTP. We kick off the CO2 we just added. So the CO2 is there very briefly. We add it and then we kick it off. And now we have our phosphate group to remake our PEP. So 2 steps there to go from pyruvate to phosphoenopyruvate, with our intermediate being oxaloacetate. So that's step 1 of gluconeogenesis. And this is only done because we have certain enzymes that are very negative, irreversible. All right. Now, before moving on the pathway, we have to dig a little deeper into this first reaction. And the reason for this is where these reactions happen inside the cell. So the first step of making oxaloacetate is in the mitochondria. However, to make PEP all the way to glucose, that's in the cytosol. And you remember glycolysis, Glycolysis happens in the cytosol. And since gluconeogenesis uses the same enzymes, it makes sense that happens in the cytosol as well. So what we need to do is we need to be able to move this oxaloacetate out of the mitochondria and move it into the cytosol. However, it's not that simple because oxaloacetate does not have a transporter. We do not have anything inside of our cells to move oxaloacetate from the mitochondria to the cytoplasm. So we have to do a bunch of chemical trickery, some biochemistry, to change oxaloacetate in the mitochondria and then change it back in the cytosol. What I mean by that is we need to convert oxaloacetate to malate or oxaloacetate to aspartate. Once it's either in the malate or aspartate form, we have transporters for malate and aspartate. So we can move that across. Once that's across, though, we can convert it back to oxaloacetate because we have the same enzymes on both sides of our mitochondria membrane to do this. So we simply, once malate's back in the cytosol, change it back to oxaloacetate, change that to PEP, and we're good to go. One other reason that we would want to do this, and I'm talking about Route 2 specifically, what Route 2 also does for us is that it transfers NADH from the mitochondria to the cytoplasm, right? Because in doing this, you spend one NADH, and in the cytosol, you gain one NADH. So in essence, not only does this transport oxaloacetate, it also transports NADH. And we learned already that NADH is very important when we were talking about glycolysis, and NADH can be used in different pathways for redox reactions. So this is just a way to move NADH from the mitochondria to the cytosol. Or you can actually run this in reverse and do the reverse. That's unlikely, although theoretically possible. So I've been talking about Route 2, and that's because Route 1 is not really used all that much. I believe I have that in the next slide. Let me erase all this. Yeah, so Route 1, we don't move NADH. So we normally don't use Route 1 because we use that NADH for gluconeogenesis. So if we want to make glucose, we have to get oxaloacetate out of the mitochondria. We have to get NADH out of the mitochondria. Route 1 does that. If you're using lactate instead of pyruvate, either way works because you can make NADH from lactate going to pyruvate directly. So that's an advantage of lactate, right? You can use either route because lactate to pyruvate regenerates NADH. But yeah, any questions so far about our first step of gluconeogenesis or our pathway system here for moving metabolites from the mitochondria to the cytosol? Can I repeat what happens in route 1? Sure. So route 1 is much simpler. You change oxaloacetate to aspartate, then aspartate can go directly back to oxaloacetate. The negative about route 1 is no movement of NADH. Because when we're doing gluconeogenesis, we need NADH in the cytosol. Gluconeogenesis. So if we start with pyruvate, we have to do route 2 to move NADH. If we start with lactate, you can do either route because going from lactate to pyruvate, that is done in the cytoplasm and it generates NADH. So you are already generating the cytoplasmic NADH you need to run gluconeogenesis if starting with lactate. So route 1 and route 2 is good for that. If already started from pyruvate, you need to do route 2 for the most part. Any other questions? All right. So we covered step 1, going from pyruvate to PEP. Then all of this is the same until we get to PFK. Remember, PFK is the major regulatory point of glycolysis, and it's irreversible. So we need fructose bisphosphatase to remove a phosphate from F16BP to make fructose 6-phosphate. Once we do that, same mechanism to go glucose 6-phosphate, and then a second phosphatase to remove a phosphate to make glucose. And this is glucose 6-phosphatase. Same enzyme that we saw with glycogen when we're going glucose 6-phosphate to glucose. This is only done in your livers and kidneys because in your liver and kidneys, that's the only place where this gene is being actively expressed. Again, if you haven't learned from glycolysis, please look after your liver. For this whole process, it costs us 6 ATPs. We use an ATP here. We use a GTP, which is the equivalent of an ATP here, and an ATP here. So 1 pyruvate is 3 ATPs. However, remember that a pyruvate is only 3 carbons. A glucose is 6 carbons. So to make glucose, you need 2 pyruvates for every 1 glucose. And that's a total of 6 ATP there. Compare this with what you get from glycolysis. You only get 2 ATP back once you count in all the spending, right? So you get 2 ATP here, 2 ATP here. And remember, this is 2 because we're running each of these twice for each glucose, because once we get to adolase, we're going to 3 carbon compounds, and you have 2 3-carbon compounds from 1 glucose. And you spend ATP there, minus ATP, minus ATP. So if you do nothing but break down glucose and then rebuild it back to glucose. So if you do this in a giant circle, where you just go like that, back up, you spend 4 ATP every time you do that, which is not good. It is possible, though, to do this, and if you need intermediates, the example we know best is pentose phosphate pathway. So let's say we need to make a lot of nucleotides. We need a lot of FADH2. You could run this type of futile cycle just to get intermediates that you keep funneling off into the pentose phosphate pathway. So it can be actually worthwhile depending on what the cell wants to do. But in general, you're talking about a loss of 4 ATP per complete cycle. So here's a review. We're going to skip question 8 because that was on test 1. What triggers glycogen breakdown muscle in the liver? Make sure you review that for the final exam, but I won't be asking you that on test 2. That was test 1 information. What we are going to continue to talk about, though, are substrate cycles. So this is fructose 2,6 bisphosphate down here. And this is liver FBPase 2. HCl goes in, phosphate comes off. So that's what's written at the bottom there. That's probably cut off for you. And we talked about substrate cycles already a little bit when we talked about the idea of glycolysis. And I was mentioning, this happens with gluconeogenesis. We'll talk about it then. So now it's the time to talk about it. There are 3 different substrate cycles that are regulated. And they are the 3 places where you have enzymes that are very large. So reaction 1 of glycolysis, reaction 3 of glycolysis, reaction 10 of glycolysis. It's where our cycles are going to be. And flux between glycolysis and gluconeogenesis, it's going to be based on PFK, FBPase. Makes sense, right? PFK, we already know that PFK is the main determiner of flux of glycolysis. It is the enzyme responsible for turning on and turning off glycolysis. So that's why it's our control point for gluconeogenesis as well. And the way that these 2 enzymes are regulated is actually quite clever. And at a very global level, you can regulate this flux by how much of each version of enzyme is present. But that doesn't work for this system, because these are actually the same enzyme. If you were to look at a picture of this, I'm going to draw it very crudely, but one part of your enzyme is FBPase, and then connected to that same enzyme is another domain, which is PFK. So same enzyme, but each part of the enzyme does the opposite function. One part does glycolysis, one part does gluconeogenesis. And these can be regulated by PFK2 and FBPase2. And they can also be regulated by a ton of allosteric effects. phosphorylation by pKa, phosphoprotein phosphatase, PP1 that we talked about. So phosphorylation regulation, and also regulation by fructose 2,6 bisphosphatase, which is right here. So fructose 2,6 bisphosphatase is not in glycolysis. And what we do is that, excuse me, is that we make it from fructose 6-phosphate. So we take F6P, and if PFK2 is turned on, we add a phosphate to F6P to make F26P. However, if FBPase2 is turned on, we do the reverse. So these names are actually nice because it's telling you which side it's turning on. So PFK2 makes fructose 2,6-phosphatase. That's from fructose 6-phosphate. While FBPase2, that destroys fructose 2,6-phosphate. Fructose 2,6-phosphate turns on PFK, turns off FBPase. So we control this enzyme that's made out of two different parts. One way we can control it is by fructose 2,6-bisphosphate. If we have that around, PFK, you're turned on, FBPase, you're turned off. If we don't have that around, the opposite happens. The amount of this molecule that is inside your cell is determined by PFK2, which makes it, and FBPase2, which destroys it. And that's how we mainly control flux, through glycolysis and gluconeogenesis. Right. So any questions about that information? Any confusion? All right, if you're still typing, feel free to type. But since I'm not seeing anything right now, I will continue on. Yeah, so we have a question here. The growth of new blood vesicles called angiogenesis that can be triggered by a lack of oxygen. It involves the rapid proliferation of endothelial cells, which make your blood vessels. These cells make high levels of PFK2. Why would it make sense for new blood vessels to have high levels of PFK2? Then I'll actually just go ahead and answer this one. So one place we see this is with cancer cells, right? Cancer cells are trying to make blood vessels, new blood vessels all the time. And the reason why, if you are doing angiogenesis, the reason why you want to make PFK2, because this turns on glycolysis. So you are rapidly creating cells So you need a lot of energy to support this rapid creation. And with glycolysis, you don't need oxygen because you can just take pyruvate and make it into lactate and keep glycolysis going. So it kind of makes sense. If you are in a condition as a tissue where you are not getting oxygen, you need to get oxygen fast. So you want to make new blood vessels super fast to try and get that oxygen. And to make new tissues or new blood vessels, rather, you need a lot of energy. So to fuel that energy, we are going to run glycolysis. And to make sure glycolysis turns on, we need to make sure PFK is turned on, because PFK is the major control point for glycolysis. To make sure PFK is turned on, we need F26P. To make F26P, we need PFK2. So you can see how all the dominoes kind of just fall in place when you look at the big picture of what's happening, why are we doing this, and then you focus smaller and smaller and smaller and smaller into the cellular components of what's going on there. So any questions about that explanation? Did I lose you? Was that didn't make sense? All right. And as always, if you are still typing, feel free to continue. So there are other ways we can control flux through gluconeogenesis. This is alanine. This is pyruvate. Pyruvate kinase, we can actually stop that, the last step of glycolysis, by alanine. Alanine can be converted to pyruvate. So if we have a lot of alanine around, we can actually use that to go and make glucose for us. And the way we do that is through a transamination, which is something we're going to look at a lot when it comes to fatty acids. But a transamination simply means we're going to move our amine. We take an alpha keto acid. What an alpha keto acid is, it's basically an amino acid without the amine group. And we make an amino acid and we get pyruvate, which technically you could call an alpha keto acid, right? So in case you haven't really noticed before, the only difference between alanine and pyruvate is this: instead of an NH3 group here, we have a double bond O. Otherwise, they look exactly the same. We can also affect flux through enzymatic changes. So the amount of enzymes you have, that's on a DNA level. That's slow, but is long lasting. So changes in amount of proteins you have, that takes about 40 minutes to kick into effect. And that's really turned on by your hormones, right? Different hormones you release will turn on and stabilize or destabilize mRNAs. And eventually you'll get that. For example, insulin will, that's actually PPCK, not PEC, will inhibit PPCK, which makes sense, right? Insulin means have glucose, while PPCK makes glucose from pyruvate. We talked about that today already. So if you have insulin around, why would you waste energy in making glucose? You have glucose in your blood already, so don't make any more of this PPCK. while cyclic AMP, that means you are very low in energy and you need glucose for energy for vital organs like the brain. So what can happen like in your liver is that the liver will say, okay, the body's low energy. I need to start making glucose here. So turn on PPCK, turn on FBPase, turn on glucose 6-phosphatase, or rather make more of it. Start getting that glucose out into the blood. On a more short term, you can do phosphorylation. And we talked a lot about this 2 weeks ago now, where we looked at how phosphorylation is a very big player in the pathway of all these systems. So again, very complex of how all these systems are related. But alanine, surprisingly enough, is also a major player when it comes to gluconeogenesis. And that is the end of the slideshow we started 2 weeks ago. Hey, we did it, everybody. I'm going to switch to the new slideshow in test 2 folder. So I'll probably make another announcement about this. But even though this is in the test 1 folder, and what I might actually do is I might just actually put these slides into the test 2 folder. Everything passed here in our test 2 folder. We have a question. So transamination just takes away a amine group to create pyruvate. In the example shown here, that is what transamination is doing, is that is taking the amine group from alanine, putting it onto something else, and you're left with pyruvate. Transamination in the big scale is just anytime you move an amine group. So that's this reaction to transamination, but not all transaminations make pyruvate. And we'll see in fatty acids how that's the case. Any other questions before I move on to new PowerPoint? Okay, so the group gets completely, so the question, okay, so the group gets completely removed or is just moved to a different spot. It is, so both, in that it is removed from alanine completely to make pyruvate. So here the amine is removed if we're going from left to right. However, it is being moved to this alpha keto acid. So this alpha keto acid, let me get the right color. This new amino acid has the NH3 from alanine, right? So we took something that didn't have an NH3. I guess it would be COOH probably. And we made it an amino acid using alanine's NH3. So that's where it went. But in the in what we're talking about for making glucose, for us, if we're just concerned on the glucose pathway, it disappears forever, never to be seen again. That makes sense? Yeah, no worries. And we'll see how that actually turns into urea and we pee it out. That's ultimately where that NH3 goes. You excrete it from your body. All right. So let's see here. Let's see if I can do this with my new screen without messing up too much. So if you cannot see my screen, let me know, because I had that problem yesterday. But if you can see my screen, fantastic. What we are talking about today, well, the rest of today, we went from glycogen to glucose. Let me get out of red because red is angry. We went from glucose to pyruvate. We went from pyruvate to lactate. We went from glycolysis to phosphopentose pathway. We just looked how to go backwards, I guess, to glucose. Drew my arrow a little big there. Now we move on to our next big cycle, the citric acid cycle. Let's look at the final fate of pyruvate. And for oxygen breathing organisms like us, you can think of the citric acid cycle as just a continuation of glycolysis. Hopefully you have an appreciation after this last month that it's not so simple. You have a lot of different pathways you can go through, but in the big picture, yeah, it's a continuation. The big idea here is that we're going to take this 3-carbon compound and reduce it to carbon dioxide. That's why we breathe out carbon dioxide, one of the reasons, because everything we eat, we change it into carbon dioxide and water. All right, so we're going to break it down into carbon dioxide. We're going to get electrons, and we're going to use that electrons to make ATP. Right now, we're only going to talk about pyruvate entering the citric acid cycle. Fatty acids, this is their fate as well. Amino acids, when we use those for energy, that's their fate as well. So no matter what you eat, I'm not a nutritionist, but it's always funny to me when I see people like, don't eat sugar or don't eat carbohydrates, only eat protein to lose weight. And then you look at the biochemical scale and it's like, it all goes to the same place. It all goes to acetyl-CoA, right? If you eat fat, if you eat pyruvate or carbs, or if you eat protein, it all goes to the same place, acetyl-coenzyme A, and then through the citric acid cycle, right? So we're going to look at the citric acid cycle now. And we have a bunch of reactions. Yay. This is our new glycolysis. If you're wondering how I'm going to ask you questions on the citric acid cycle, it's going to be very much like test 1 for glycolysis. So you can expect the same thing. 8 reactions here. Every time you go around the circle once, you create 2 carbon dioxide, 3 NADH, FADH2, ATP, GTP. So you are getting a ton of energy just by going around the cycle once. Acetyl-CoA is 2 carbons, right? So 1, 2. So 2 carbons go in, 2 CO2s come out. So that's where all the carbon from glycolysis is going, directly into carbon dioxide. Again, we talked about not just pyruvate, a lot of places. This is all done in the mitochondria. This is why it's the powerhouse of your cell. Note that this carbon dioxide that we breathe out it's not from these carbons. What we will see in the first step, we take our acetyl-CoA and we combine it with oxaloacetate, our little friend oxaloacetate we just talked about. And it's the carbons from oxaloacetate that get changed into CO2. And they're nicely labeled, actually. So you can see this star carbon dioxide or carboxylic acid comes off right there. And it is this group, I believe. that gets another oxygen added to it and removed as carbon dioxide. So the carbons are coming from oxyl acetate, but this acetyl-CoA is eventually turning into oxyl acetate, so it takes 3 rounds. So these 2 carbons will go 3 rounds before they are expelled as carbon dioxide. A lot of intermediates here that are important for a lot of different things. Alpha-ketoglutarate being a big one, citrate, succinate, fumarate, oxyl acetate. We'll see these in different pathways, malate as well. We just looked at that in the malate shuttle, right? So these things are used for a lot of different things. And in total, we get 4 pairs of electrons, which means 8 electrons. So remember, NADH, FADH2, they each carry 2 electrons. So that's where our 8 electrons are coming from. So that's the big overview of the citric acid cycle. And so before we go into looking at the actual reactions, any questions about the overview of the citric acid cycle? Why is it ATP or GTP? Different enzymes. Logically, I don't know why. It's just that you have certain enzymes in your cell that will only use GTP or ATP. And succinyl-CoA synthetase, for whatever reason on the evolutionary scale, decided to use GTP. I don't know why that is. So I don't have a better answer for that, unfortunately. Keep in mind, GTP has has like the same energy as ATP. The only difference is that you spend them in different places. So I guess you can think of GTP as the euro and ATP as the dollar. Different enzymes accept different currency. Or I guess in Texas, it should be the peso, but yeah, euro is the first thing I thought of. Yep. So unfortunately, I don't have a better answer to that other than it just does use those. All right. So question 1, I guess I'll answer this as well, just for sake of time here. So we know pyruvate has other fates. So why would we want to run the citric? When would we run the citric acid cycle? Okay. So the other fate of pyruvate that we focus on was going to lactate and making glucose. So those are the 3 fates, really. There are more, but these are the 3 ones. Pyruvate goes to glucose, pyruvate goes to lactate, pyruvate goes to citric acid cycle. So why would we ever do this pathway? Another way to answer that is, why would we not do these 2 pathways? Right. So for this to be the case, we need to have oxygen around. Remember, if we don't have oxygen, we don't run the citric acid cycle, and we go from pyruvate to lactate. So we need oxygen. We have to be low in energy too. If we're high in energy, what happens is that we're going to take our pyruvate and make glucose from it. So you need to have oxygen and you need to be low in energy. Then you start to run the citric acid cycle. That's when we will do it. Glycolysis, well, we already talked about this today. That's the cytoplasm. Well, this happens in the mitochondria, which means we need to get the pyruvate we made in the cytoplasm and transfer it across the mitochondrial membrane so it can interact with the citric acid cycle. So we're going to be looking at transporters here at some point to see how this is done. So make sure you keep all that, the big picture, as we go and look at the individual enzymes, make sure you keep the big picture in mind of like when we would run this and what does this mean on the cellular location of things. That makes sense to people, or any questions about that? All right. And two, originally this question was scheduled if we didn't just talk about this, but oxaloacetate. We just talked about that about almost an hour ago now, right? That is an intermediate for making glucose. Does anyone remember the other thing we talked about? And it was very briefly we talked about it, but there was another intermediate that interacted with PFK. Remember what that was? And it was like a month ago now, so no worries if you don't. That'll be citrate. So citrate inhibits glycolysis. We'll look at this in depth a little bit, but citrate will go and inhibit glycolysis. Oxaloacetate can go and make glucose. So just examples there of different intermediates being used. We haven't talked about it yet, alpha ketoglutarate. That's really good at moving NH4 groups. That's mainly used in fatty acid, protein, biochemistry, metabolism. But we really haven't talked about that one yet. All right. So before we get into the citric acid cycle, we have like a precursor step, because the very first molecule that moves into citric acid cycle is acetyl-CoA. However, if we're talking directly from glycolysis, the last molecule in glycolysis is pyruvate. So what we need to do is convert pyruvate to acetyl-CoA, and this is done by the enzyme pyruvate dehydrogenase. And pyruvate dehydrogenase, when we talk about it, it's going to be made out of 3 enzymes. We're going to call them E1, E2, and E3. These aren't connected by covalent bonds. There's nothing holding them together other than non-covalent interactions. So you got your hydrogen bonds, your hydrophobic interactions, electrostatic interactions. So amino acids not in bonds holding these together. And this is kind of what it looks like. And this picture is a little confusing, so let me break it down. What happens, I believe this is E2, is that you have 3 enzymes. You have green, which is E2. I think, I'm going to cheat and look at the next slide. Yeah, okay. So this is E3, this is E1. And here's them separated, A and B is it's separated. and C is when you combine these two pictures, you get C. So A picture plus B picture equals C picture, right? And they're arranged in this cube-like formation so that when pyruvate, I'm going to say that as like P, will go and interact with enzyme 1 first, it can immediately go to enzyme 2 because it's right next to it. So by an advantage by having all these enzymes close to each other is that your substrates don't have to travel far and the reaction goes faster. This is different than glycolysis and basically different than everything we talked about, because in glycolysis, all the enzymes are not connected. They're just floating around inside your cytoplasm, and these substrates have to go and find them. Here, everything's like connected together in this one giant moving cube. So the substrates don't have to move far. That means the intermediates don't have a chance to interact with anything other than the enzymes. So like no chance of any side reactions. You're only going to do the main reactions. And you can actually control this very easily. Since everything's in one location, you can control if this runs or not, right? So control, you have a very high level of control by having all your enzymes close together. And this is actually the biggest multi-enzyme complex that we know of. What I'm showing here, this is the E. coli version, the most well-studied version. Mammals, it's a little more complex than this. So we're just going to focus how this happens in E. coli. Like I said, well-studied, and it gets the gist of what happens down. So that is how our pyruvate dehydrogenase complex is organized. 3 enzymes in a giant cube that are all next to each other. So let's actually look at the reactions for this. So our first reaction, enzyme 1. We take our pyruvate, chop off the carbon dioxide. This is extremely similar to the reactions we saw in yeast where we create ethanol from pyruvate. In that reaction, if we remember back, we chopped off carbon dioxide to make ethanol. Here, we're chopping off carbon dioxide, sticking it on to this TPP. TPP is simply, it's a cofactor. It's just like it's part of the enzyme complex. But all we're doing here is we're sticking our 2 carbon unit onto TPP to make our hydroxyethyl. So if you remember from organic chemistry, ethyl means 2 carbon, hydroxy, OH, hydroxyethyl, TPP. So that's our very first reaction. And it's done. That's what E1 does. That's all that E1 does. After we have attached our pyruvate to E1, the next reaction, number 2, is that we're going to take this hydroxyethyl group and we're going to give it to lipoamide. This is done by E2. By doing that, our TPP is regenerated in E1, and it's ready for another reaction. So literally all enzyme 1 does is knock off a carbon dioxide, and handed over to E2. This lipoamide group, when we look at it, it's a giant arm. So this is our lipoamide. It's a giant arm that is connected to a lysine, plus this thing's known as a lipoic acid. You don't need to know the structure of this. Don't worry about this. Just know that E2 has this large flexible arm. And the way it actually works, I have a question. Reaction, are we attaching the CO2 or the pyruvate? Oh, okay. Sorry, I must missaid my word. So in reaction 1, we are cutting off a carbon dioxide from pyruvate, and we're left with this ethyl hydroxy group. That's what we're attaching to TPP. CO2 just flies off and you breathe it out. So we are attaching pyruvate minus 1 CO2. All right, so in enzyme 2, that answer your question, by the way? That makes sense now? I guess I should ask that before I move on. Okay. So enzyme 2, we have this like large flexible arm. Think of it like a spring almost. And actually, one sec. I guess it's better to look at me while doing this. So this complex, imagine I'm enzyme 2 and I'm in the center. And to either side of me is like enzyme 1 and enzyme 3. So enzyme 1, enzyme 3, enzyme 2. What happens is, sure, my phone will work, is that pyruvate comes to enzyme 1. And then enzyme 2 takes it away. Actually, I only have 2 arms, so this is going to work too well. But oh well, I will make it work. So enzyme 2 takes pyruvate away. Attached to enzyme 2 is this long flexible arm. So that's what takes it from enzyme 1, and it will give it to enzyme 3. This long flexible arm is that lipoic acid I'm talking about. So enzyme 2, that's why it's in the center. It's doing all the work. It is moving around our substrate because it has this long, flexible arm made out of lysine in it. That's what's being shown here. So we grab our pyruvate from enzyme 1 and we put it on enzyme 2. When we do that, we break our disulfide bond. And we go and interact with coenzyme A. And the pyruvate from step 1, we attach it to coenzyme A. We make our product, acetocoenzyme A. That goes off to the citric acid cycle. However, enzyme 2 is sad because its disulfide bond is broke. And it cannot do another reaction unless its disulfide bond is reformed. But that's reaction 2 and 3. Reaction 2, take my 2 carbon from enzyme 1. Reaction 3, give it to coenzyme A. I do that via a disulfide bond. Reaction 3, already talked about it. Don't worry about that. I'm not going to ask you that mechanism. We have enough stuff to memorize. So you can just cross that out of your notes right now. So reaction 3, just talked about coenzyme A. Again, I'm not going to ask you arrow pushing on our PDH complex. So this we need to know, but not the actual arrows. All right, let's move on to reaction 4. So we moved our pyruvate 2-carbon complex to acetyl-CoA. E2 is really sad. Its disulfide bond's broken. It no longer works. So that's where its old friend E3 comes in. E3 has a disulfide bond, and it sees enzyme 2 being sad. So E3, reaction 4, will reform the disulfide bond of E2. E2 is now ready to go for another reaction, but that leaves E3 with a broken disulfide bond. So we need to fix that. So the whole purpose of E3 in this, all it's doing is it's restoring the disulfide bond in E2. That's E3's whole purpose in life. However, it can only do that once, then it needs to be regenerated. So NAD plus will take the hydrogens away from E3, reforming our disulfide bond. So overall, pyruvate comes in, CO2, I guess NAD plus pyruvate comes in, CO2 comes out, acetyl-CoA comes out. So I should add COA plus NAD plus pyruvate, and NADH comes out. This is done in a series of 5 steps laid out here. Know how the 5 steps are run. Don't need to worry about the mechanisms for this. And then we are done. E3 is reoxidized. So any questions about these 5 steps of turning pyruvate to acetyl-CoA? Did I lose anybody at some place? Was something I said confusing and you're not quite sure? Something you want to hear again? Anything at all? Can you write the 5 steps out again? I can do that very briefly. Sure. Pyruvate loses CO2 and forms TPP hydroxy ethyl, which is right here. That's done in E1. That's reaction 1. Reaction 2, lipoamide takes the hydroxyethyl to form our acetyl dihydryl lipoamide. Let me rewrite that because my handwriting is slop. Acetyl dihydrolipoamide. That's E2, and you regenerate TPP from that. E3, your coenzyme A will combine with your hydroxyethyl to make coenzyme A, acetyl coenzyme A. This is all done in E2 again. E2 disulfide is now broken. So E2 no longer works after step 3. Step 4, regenerate E2 disulfide using E3 disulfide, E3 disulfide broken. Step 5, regenerate. E3 disulfide by converting NAD plus to NADH. That is probably the simplest I can write down the 5 steps. That helps seeing it laid out that way. Any questions still? Okay. And remember, by step 3, we are done with pyruvate. Like that pyruvate's already been turned into coenzyme A. Step 4 and step 5 are simply regenerating our enzymes. All right, and I believe mechanism review, I actually just did that, so no problem. And unfortunately, if we were in live class, this is where I have a fun little game where we break into, well, I don't know if it's fun, but we have a little game where we break into groups and we have like models and you become different enzymes, but we don't have that because COVID. So unfortunate. But at this time, it is 1:45. So I will call it here. We will look at the citric acid cycle Tuesday and dive deep into that now. I will put a short homework up. I know it's been a while since we've done one of those, so don't forget about that. Otherwise, if you have questions about anything, never hesitate to reach out to me. I will answer those. And for that, have a good weekend. Welcome back, and hopefully I'll see you on Tuesday. Take care, everybody.